Ross Granville Harrison, January 13, 1870, September 30, 1959, was an American biologist and anatomist credited as the first to successfully grow artificial tissue culture. His work also contributed to the understanding of embryonic development. Harrison studied in many places around the world and made a career as a university professor. He was also a member of many learned societies and received several awards for his contributions to anatomy and biology. Harrison successfully cultured frog neuroblasts in a lymph medium, proving that nerve fibers develop without a pre-existing bridge or chain and that tissues can be grown outside of the body. He published the results of his studies in 1907. This part of Harrison's research was the first step toward current research on precursor and stem cells. While Harrison himself didn't develop this area of research any further, he encouraged others to. He was considered for a Nobel Prize for his work on nerve cell outgrowth, which helped form the modern functional understanding of the nervous system, and he contributed to surgical tissue transplant technique. In 1891, he participated in a marine zoology field trip to the Chesapeake Zoological Laboratory in Jamaica. Attracted to the work of Moritz Nussbaum, he worked in Bonn, Germany during 1892 to 1893, 1895 to 1896, and 1898 and became an MD there in 1899. Harrison gained his Ph.D. in 1894 after courses in physiology with H. Newell Martin and morphology with William Keith Brooks. He devoted study to mathematics, astronomy and also the Latin and Greek classics.
During the First World War, Harrison studied embryology and the symmetries of development. By means of the dissection of embryos followed by transplantation and rotation of the limb bud, he demonstrated that the main axes of the developing limb are determined independently and at slightly different times. Determination of the anteroposterior, anterior-posterior, axis preceding that of the dorsoventral dorsal-ventral axis. Harrison dissected Ambystoma punctatum salamander embryos and transplanted limb buds to determine whether the limbs developed independently or according to instructions from host cells. When the limb buds were transplanted in halves or doubled, they still developed into normal limbs. Harrison concluded that the information from the host, the surrounding embryo cells, directed the cells to develop normally even though they were transplanted in halves or doubles. Therefore, the limb buds were all equipotential, meaning they all developed the same way, and the tissue around the limb buds determined their dorsoventral orientation. However, when a left limb was put on the right side of the body, a left limb grew anyway despite its relocation. The same pattern occurred when a right disc was placed on the left side of the body, a right limb grew. Harrison also transplanted inverted limbs. When a left disc was inverted, it grew a right limb, and vice versa. Harrison then concluded by this data that the buds determined anteroposterior orientation independent of the surrounding host tissue. Harrison's research led him to assume that the development of limbs is not determined exclusively by the limb buds or by the environment, but that both of these factors influence how an embryo develops. Harrison published the results of this study in 1921 in the Journal of Experimental Zoology in a paper titled, On Relations of Symmetry in Transplanted Limbs.